Thank you for the kind introduction and for giving me the opportunity to be here presenting this work today. Good morning, my name is Teresa I López and I'm currently doing my PhD in doctoral task group, searching for new biomarkers of ALS. Today I bring you here the first results of my thesis that I have compiled in a poster entitled Expression Study of a Cluster of 5 non-encoding RNAs in a mouse model of amyotrophic lateral sclerosis. First of all, I'm going to start doing a brief introduction of what non-encoding RNAs are, since they are relatively recently discovered molecules, and maybe for some of you may be unfamiliar. As their name suggests, non-encoding RNAs are RNA molecules of more than 200 nucleotides in length, which are not translated into proteins. They can be classified into circular RNAs and linear RNAs. These last are the one I focus my work on, and they can be further divided into intergenic, intronic, sense-overlapping, or antisense RNAs. What have we to know about non-encoding RNAs? They are present in a large diversity of species, including animals, plants, yeast, prokaryotes, and even in some viruses, but they are usually low expressed. That's why they have traditionally been considered as a kind of transcriptional noise, but nowadays there is more and more evidence that they play a significant role in a wide variety of important biological processes, such as the RNA metabolisms. In fact, they can act as transcriptional, translational, and epigenetic regulators of a wide diversity of genes, and also in other processes like proteostasis and autophagy, motor neuron development and differentiation, apoptosis, cell co-regulation, inflammation, and neuronal and muscular atrophy. Also, these processes have been, let's say, being detected like altered or misregulated in ALS. We thought that this kind of molecules could be good potential biomarkers for ALS. As you probably know, because these days we have been listening to talks about amyotrophic lateral sclerosis, this is one of the most common neurodegenerative diseases. It is characterized by the progressive and selective loss of motor neurons from the spinal cord and brain, which leads to atrophy and paralysis of voluntary muscles. It is a devastating disease with no cure that results in patient death within 3 to 5 years from diagnosis. In fact, one of the main challenges that it poses is this late diagnosis that is due to the lack of good biomarkers. So with this background, we set the aim of this study, that is to unravel the expression profile of 5 non-encoding RNAs in the main tissues affected in ALS. That is to say, the nervous system, and particularly in the spinal cord and muscle, that specifically will look at the quadriceps along the different stages of the disease using a mouse model of ALS. So for this purpose, we collected samples from 24 mice balanced on sex and genotype, half of them being have wild type and half of them having a mutation in the SOD1 gene. We choose this mouse model because it's one of the best characterized, let's say, for this disease. And we did this at 3 different time points. The first one could be the 60 days, which represents a presymptomatic stage, also at 100 days that could match with an early symptomatic stage in this animal model, and 120 days that would represent a late symptomatic stage. We took the samples, we started RNA, and we also did a bibliographic search for potential biomarker candidates that has been previously reported in molecular mechanisms associated with neurodegeneration. We did the proof of design and the validation of the amplification product by sequencing, and later we did the analysis by real-time PCR. Results show significant expression differences between SOD1 and wild-type males in these mice as soon as 60 days. If you look here in females, we cannot see any differences. Probably this is due to the fact that females in this mouse model usually develop the disease later than males. So they are always like with a kind of retard. So at 105 days, that would be the early symptomatic stage, we still see some of these differences. We can see it now also in females. But interestingly, at late symptomatic stage, all these differences disappear. And London coding 5, in which no significant differences in expression had been detected until now, becomes significantly increased in both the spinal cord and muscle of males and females. Temporal patterns of expression for each London coding are likely related to different processes that are relevant at distinct stages of the disease. We also we were interested also in testing whether the levels of these London coding 3 and 5 could correlate with weight. Why London coding 3 and 5? Because London coding 3 has shown a large variety of expression between individuals, and London coding 5 was misregulated at the latest stage. That is when we weighed the animals. So indeed, we found that in both cases, there was a correlation of weight with expression levels in muscle. This result is remarkable because weight is associated to life expectancy in this disease. In summary, we can say that non-coding RNAs were differentially expressed in SOD1 and wild-type mice at different ages, and this expression profile not only varied according to age, but also according to sex and tissue. These findings are consistent with the different disease progression observed in males and females of this specific modern model of ILF, as well as its different frequency in patients. Among all the London coding studies, London coding 1, London coding 3, and London coding 5 stand out. London coding 1 levels are decreased at earlier stages in a spinal cord of male and female SOD1 mice. Likewise, London coding 3 is diminished at earlier stages in a spinal cord of both male and female ILS mouse model and in quadriceps of male SOD1 mice. Last but not least, London coding 5 is increased at later stages of the disease in a spinal cord and muscle of male and female SOD1 mice. It shows a consistent progression throughout the whole disease course, standing out as a possible candidate for prognostic or diagnostic biomarker of this disease. Furthermore, London coding 3 and London coding 5 expression level in muscle of later stage mice negatively correlate to weight. Finally, altered expression patterns of the London coding RNAs studied suggest that they may play an important role in the pathogenesis and development of this disease. What we want to do next? Well, we want to study To extend the study to other non-coding RNAs and test the expression of the most relevant candidates in serum. And we would also like to correlate the levels of the most promising candidates with the survival of SOD1 animals in serial blood samples or biopsy. I would like to end by thanking all the members of the group, as well as Dr. Alberto García Redondo from the Instituto de Investigación Sanitaria Hospital 12 de Octubre, who also participates in this project. And of course, to all the funders who have made this work possible. Thank you, that's all. And if you have any questions later, I will be happy to answer them.